everyone. I'm back after a couple of months of absence, and I'm looking forward to interacting with all of you today. I see some familiar names out there. So today what I have planned is that I have a short presentation, and I hope that you guys will enjoy it because this is a topic that I have been mulling over for quite some time as to what determines hunger and satiety. What are the signals we have to eat and control the amount of food that we eat? At the end of this presentation, I hope to have a quiz, and hopefully in the quiz, some of you will give me answers that will help me decide whether some of the inferences I have made from looking at the literature are correct or not. So the topic for today is the regulation of hunger and satiety. And at the outset, this is my perception, and I have been talking about this for quite some time, that humans are programmed to overeat. Because overeating created a survival advantage in most of human history. The availability of food was not constant, and the presence of a supermarket, a personal fridge, and pantry is just a blip in time, and humans have not had the time to evolve to this kind of presence of food. So hunger and satiety are perhaps related to many factors, but for the purposes of today's presentation, I'm going to focus on energy-related food intake. In other words, you're energy deficient, the body makes you eat. So this is like a homeostatic system that we have in the brain. Then there is the hedonic or pleasure-seeking food intake. Hedonic means pleasure, something that creates pleasure in us. And then there are hunger and satiety hormones that are present in the gut, and we will kind of look into that. And then finally, I want to evaluate and see how taste receptors, we have several taste receptors, and we're going to go through that, how these taste receptors interact with our food intake. So this is explaining the homeostatic or energy need based food intake. And this is present in a portion of our brain, which is called the hypothalamus, and it's in the arcuate nucleus. So this nucleus is driving the energy-based homeostatic brain center for appetite regulation. This center elaborates certain orexigenic stimuli. Orexigenic meaning stimuli that promote hunger. There are certain peptides, neuropeptide Y, and agouti-related protein that promotes the feeding behavior. On the other hand, if you are energy sufficient, in other words, this promotes feeding behavior, and then this feeding behavior will, through certain mechanisms, activate propio-melanocortin, and I think the spelling of that is wrong. And this creates an anorexigenic stimuli. So anorexigenic stimuli will prevent you from then consuming food because there is not a need to eat food. And it would have been beautiful if all our food intake was based on energy needs. In other words, the homeostatic center drove our food intake based on, hey, we need energy, eat food. We don't eat energy, we stop food when energy is sufficient. But fortunately or unfortunately, that's not the case, because there is a hedonic center, a pleasure-seeking center in our brain, and this resides in an area in our brain called the nucleus accumbens. And this is a dopaminergic system. In other words, activation of the system creates a dopamine response, and dopamine is a pleasure signal. And this pleasure signal from here then projects to different parts of the brain, especially the frontal cortex. And it says, this food gave me pleasure. Not just gave me energy, but gave me pleasure. Let's do it again. So it creates a kind of a habitual response. And some of the foods that activate the pleasure or the hedonic center is, of course, sugar and fructose, because they induce a dopamine release. And it's also been shown that withdrawal of these substances, withdrawal after exposure of sugar and fructose, leads not only to cravings in the short term, but there is persistence of cravings in the long term. So all of us who have had the pleasure of eating sugar are still on the hook on getting drawn into consuming these products again. So the low carb community should be aware that relapses are certainly in our genetic makeup, in our physiological makeup. Now, the hedonic or pleasure-seeking centers are not only activated by sugar, but they are also activated by sweeteners. So low-calorie sweeteners that don't have any calories activate dopamine release. In addition, withdrawal of these sweeteners will also create cravings, and these cravings, just like sugar, can persist. So here is a molecule of sucrose or table sugar, as it's shown here. It is a covalent bond. It is linkage of two molecules, fructose, which is a type of sugar, and glucose, which is another type of sugar. On the other hand, this is glucose. Glucose is different. It looks similar to sucrose when you see it, but perhaps it's very different than sucrose because it elicits less of a sweet response. It doesn't give the same degree of sweet taste. On the other hand, fruit sugar, fructose, is perhaps sweeter or as sweet as sucrose is. So these are very important differences that we need to consider, and not everybody is aware of it. Most of you are. So if you look at the homeostatic energy-based center in our brain that is driving food intake, even if that is exposed to fructose or sucrose, it drives the orexogenic stimuli. In other words, it drives the stimuli that promotes feeding behavior much more than what would be needed based on the energy needs. So in other words, even though this is a homeostatic center, it can be overdriven by certain food components that would promote feeding behavior. On the other hand, glucose, which is found like in starch, like potatoes, does not have the same drive to stimulating the orexogenic stimuli, in other words, feeding behavior. On the other hand, through certain peptides, these are satiety peptides that we will learn a little bit, it can actually drive the anorexigenic stimuli that will cause a cessation or a reduction in food intake. So glucose is very different than sucrose or table sugar and fructose. So now we've looked at the brain centers that regulate energy needs and brain centers that provide hedonic or pleasure-seeking food intake. But now we're going to move on and look at hunger and satiety hormones in the gut. So our gut, the stomach, the small intestines, and large intestines elaborate certain hormones that regulate our hunger and our satiety. So among the hunger hormones is ghrelin. Ghrelin is elaborated by the stomach and it drives food intake. It drives gastric emptying. It improves peristalsis. So ghrelin is an important hormone that is driving our food intake in addition to another hormone, which we will see in the next slide called motilin. On the other hand, the small intestines and colon, and to a certain extent the duodenum, elaborate certain hormones like cholecystokinin, peptide YY, and GLP-1. And these slow down the gastric emptying. They have other functions like bile production and pancreatic secretions. But for our purposes, they have a satiety effect because they're slowing down gastric secretions, reducing motility. And all these three compounds, although the arrows are going only from PYY and GLP, even from cholecystokinin, there are signals that are going through the bloodstream to the hypothalamus to promote satiety and reduce food intake. So these are the hunger hormones, the hunger promoting hormones, the ghrelin, which is a 28 amino acid peptide elaborated by the stomach, and the motilin, which is a hormone that is produced not only by the stomach, but also by small intestines and colon. And it causes stomach contractions and peristalsis that promotes gastric emptying, creates hunger pangs, and creates the kind of gastric motility that will tell you that you are hungry. On the other hand, GLP, which is a representative hormone, but it also includes CCK or cholecystokinin and PYY, and these are satiety hormones. Not only do they reduce gastric emptying, they have many other functions. They affect the fat tissue and promote fat burning and reduce fat storage. They reduce glucose output from the liver, so they reduce the amount of high sugar after eating. They have a significant impact on the brain in the sense that they go and tell the brain centers to reduce appetite. So here is cholecystokinin, and it has effects on the gallbladder in terms of gallbladder emptying, on the pancreas in terms of secretion and augmentations of pancreatic juices that aid in digestion. But then also it has effects on stomach in terms of delaying gastric emptying, and on the brain in terms of induction of satiety. So in order to promote satiety, one should design mechanisms that reduce ghrelin and motilin, and design mechanisms that increase GLP, PYY, and cholecystokinin. So let's get to the fourth aspect now. Now we talked about the homeostatic, energy-based hunger centers in the brain, hunger and satiety centers. We talked about the hedonic centers in the brain that seek pleasure from food. We talked about the hunger and satiety hormones, and now we're going to change our focus onto the taste receptors. So there are, I think, 6 different types of tastes. Many people say there are only 5: the sweet, the umami, the sour, the bitter, the salty. But I think fat is not just textural, but I do think we can taste fat. This is debatable, however. There are different regions on the tongue. So in other words, taste or gustation is a chemical that's acting on a receptor on our tongue. And there are extra tongue receptors, taste receptors, as we shall see. And that creates a sense of taste of all these different types. Monosodium glutamate, mostly from glutamate, is umami. There's a sour taste, sweet taste, usually from sugar. I debated whether I should include chocolate in here, but because chocolate has cacao, which is bitter, But the reason I did is that most of the chocolate is laden with sugar and it has more sugar taste than bitter taste. And that's why I put it under sweet. A representative bitter taste would be caffeine, and we'll discuss this further, and of course, salt. The taste receptors that are present on the tongue have different shapes and different types. They have taste hairs and taste pores. They interact with their ligands, a ligand meaning either a sweet ligand or a bitter ligand or a umami ligand. And then there is intracellular signaling. There is signaling inside the taste receptor cells, following which there is enteroendocrine function. In other words, these cells then subsequently interact with other cells in the gut, in the fat tissue, in the brain tissue, to create certain responses. So they not only have paracrine, in other words, responses right next to them, like for example, a taste cell can be juxtaposed to an L cell that is producing GLP-1, which is a satiety hormone, or it could be interposed or juxtaposed to a ghrelin-producing cell that increases hunger. So that's the paracrine effect. There is also an enteroendocrine effect in which the taste cell is then signaling downstream to the cells that are producing GLP-1, cholecystokinin, or peptide YY, and tell us to increase satiety. So these are mammalian taste receptors. They respond to different compounds, glutamate for umami, sugars for sweet. The bitter taste, denatonium is an important compound that is used to check the taste receptors. There is PTC, which we will investigate a little bit further in terms of a group of people who are considered tasters and non-tasters. There is salt, and then of course there is sour taste. So the question that I'm posing here is that does a sweet taste receptor, now the taste receptor type 1, because it's T1R2, that is taste receptor type 1, that is a sweet taste receptor. Once it interacts with the ligand, it has intracellular signaling. In other words, calcium is increased inside the cell that leads to signaling. And this signaling through paracrine effect, through enteroendocrine effect, is increasing ghrelin, mortallin, which promotes hunger. It also increases the satiety hormones. But the question I'm asking is that, does the sweet taste increase food intake and reduce satiety? And you would expect that there is good information with this regards. And of course, there's a lot of extrapolation. But certainly we can see that it activates the hedonic centers as well as the homeostatic energy-based centers to promote food intake. So sucrose and fructose create hedonic signals, and they amplify the homeostatic energy signals in the brain and thus make us overeat. On the other hand, and I think I did most of this presentation because I wanted to focus on the bitter taste receptors and see if that's a tool that I can give you to reduce your food intake in order to promote satiety, reduce hunger, because I'm convinced that if we learn how to eat less, we will be healthier. So bitter taste receptors, when they interact with a bitter ligand, will have intracellular signaling, which is mostly influx of calcium into the cell, and also efflux of calcium from certain storages inside the cell. And these intracellular signaling will then create the paracrine and enteroendocrine response, in which for the bitter taste receptors is a reduction in ghrelin, reduction in mortallin, and an increase in the satiety hormones, which is GLP-1, CCK, and PYY. I had mentioned that the bitter taste receptors are not only present on our tongue, but they are found in various other tissues. This is a study that is looking at extraoral bitter taste receptors, and they found them in the fundus, the antrum, the duodenum, the colon, the tongue. But they're also present in adipose tissue. They're also present in the brain. And in this study, they gave healthy volunteers the experience of bitter taste, and they found that it decreased the intake of food because it impairs the relaxation of the stomach. You feel like you have a full stomach, so it reduces the amount of food that you eat. It increases satiety and consequently interferes with hunger signaling. So perhaps we can, as a low-carb community, now begin to think not just of fasting, but of other mechanisms to regulate our food intake. And one of them is to exploit the bitter taste receptors to our advantage. There are about 25 different human bitter taste receptors. So we just don't have one, but we have 25 different. All of these 25 different human taste receptors are shown here. I want to remove this to some other place. Maybe I put it there. And this color coding of them is indicating whether they respond just to a few ligands that are bitter tasting. So like this color indicates that they respond to more than 50 ligands that are bitter tasting. This color 20 to 50. And these are much more specific. In other words, they respond only to a few different ligands. Denatonium, benzoate, chloroquine, phenyl thiocarbamide. This is PTC, and this interacts with taste receptor 38. I put that in green, the taste receptor 38, because it requires special mention, and I'll be talking about that in a subsequent slide. Now, this study that was looking at bitter taste receptors is trying to figure out whether you can impact our food intake, improve satiety, reduce hunger, and thus impact on obesity. So this paper argues that bitter compounds, when they interact with the taste receptors, have 4 different mechanisms through which they improve our health. Number 1 is interacting with our gut. Interacting with our stomach, the small intestines, and colon, they reduce ghrelin and mortallin, which is the orexigenic hormones. So the orexigenic hormones that improve hunger are reduced. On the other hand, the satiety hormones, the anorexigenic hormones, the GLP, the PYY, and cholecystokinin, are increased. There are several papers that show this kind of a phenomenon, and consequently you reduce appetite. The second mechanism is that they work on gastrointestinal motility. They work on the smooth muscle cells that are regulating the motility of a gastrointestinal tract, and they impair the relaxation of the stomach. So in other words, if the stomach is not relaxed, it feels full, and you will reduce your food intake. It slows down gastric emptying, promotes satiety. And it causes what is called an ileal block. The intestinal mobility is reduced. The ileal block promotes food absorption and thus reduces your appetite. The taste receptors are also found in the brain, and certain bitter compounds like quinine and probably certain derivatives of coffee cross into the brain, into the appetite centers, and improve our satiety. In addition, taste receptors are found on the adipose tissue they promote lipolysis, which means that they break down fat. They promote lipogenesis, in which they prevent the storage of fat. So there is a lot of data from different studies that taste receptors, when activated by bitter compounds, can improve our health through all these mechanisms. And I wanted to explore this tool with the low carb community and give them another method through which they can improve their health. So let's talk about a few bitter compounds. Now, there are many of them, and probably 20, 25, 30 different bitter compounds that are common in our diet. But I'm going to focus on caffeine, coffee, and decaffeinated coffee, bitter melon, and cacao nibs. So cacao nibs and all of these I want to discuss. So here is bitter melon. This is a rat study. And in this rat study, bitter melon was given to the rats. It was given in a pretty high dose. And what was found is that the higher the dose of bitter melon, the more was the expression of the satiety hormone, which is GLP-1. So GLP expression was increased. Now, GLP is naturally degraded in our body through an enzyme called DPP4. This is an enzyme that destroys GLP. And by administration of bitter melon, not only did you increase GLP, but you reduce the enzyme that causes its destruction. Now, this methodology, in other words, increasing your GLP-1 and an inhibitor of DPP4 is exploited by physicians to improve diabetes. So in other words, Victoza was the first GLP-1 agonist, which is an injected form. So in other words, you have to inject it. And it provides GLP-1, which reduces gastric emptying. It reduces glucagon, so your liver is not making more glucose. And what's absent from this slide is that GLP-1 directly interacts with the brain and improves satiety. These are some of the compounds: Trulicity and Ozempic, Byetta. These are commonly used by diabetic patients, and some of you may recognize and may even be using these. And you can now understand how these work in your system. Now, DPP4 inhibitors, so these are ones that inhibit, these are oral drugs. Now, these drugs like Januvia and Tradjenta, they inhibit the enzyme that's inactivating naturally produced GLP-1. So when you use this, the amount of GLP-1 is increased, it's more available, and it promotes more of the things, the same things that is like taking Trulicity, perhaps to a lesser degree. Now, polyphenols are found in caffeine, they're found in cacao nibs. Chlorogenic acid is a derivative of coffee. This is a flavonoid, a polyphenol, which is generally found in several bitter compounds. And these polyphenols have several actions that benefit our metabolism. They increase the satiety hormones. They increase the half-life of the satiety hormones because they inhibit DPP4. The GLP-1 increases insulin secretion, so it improves blood sugar control, it improves insulin sensitivity. So consequently, there are beneficial effects of polyphenols, which are found in bitter compounds. So this is a cacao polyphenol that was used in healthy participants. And after the cacao polyphenol was given, these people were challenged with a 50-gram oral glucose tolerance test. So these people were consuming polyphenols. They were asked to drink a 50-gram solution of sugar or sucrose. And what was found is that the people who were taking polyphenol which is out here in white, had a higher increase in GLP-1 compared to the control group that did not take the polyphenol and took the sugar. The area under the curve, the total amount of GLP-1 that was produced was higher. So what that indicates is that the cacao products are improving your satiety. They're improving GLP-1 in response to food. Now, chlorogenic acid, which is a part of caffeine, increases GLP-1 production as summarized by this review, that they think that evidence that coffee increases production of GLP-1 is perhaps a mechanism that should be exploited, not just to control blood sugar, but perhaps also to improve satiety and reduce hunger. Now, this is decaffeinated coffee and other major coffee components. And what you're seeing out here is chlorogenic acid, which is found in coffee, is increasing GLP-1. Decaffeinated coffee is not having some of the same effect as the chlorogenic acid does. So I'm not sure whether there is data that is robust and solid for me to tell you that decaffeinated coffee would be just as good satiety promoting and an appetite suppressant as regular coffee will be, but I will be interested in some of the experiences that people have had. Now, we were going to talk about the specific taste receptor, bitter taste receptor called 38, because this taste receptor is polymorphic. By polymorphic, I mean it comes in different types. It comes in a type in which the amino acid sequence is PAV. In other words, homozygous. That means you inherited genes from both parents in which you are homozygous with a certain amino acid pattern. And these people are called tasters. That means they can taste bitter compounds, and that's 75% of the population approximately. And these people can taste this bitter compound called phenyl. This should be an H, phenyl thiocarbamide. Whereas homozygous with a different amino acid, which are called non-tasters, which is 25% of the population, do not taste the bitterness of this. So this is a genetic difference between us. And we need to figure out if this genetic difference is telling us whether it's an advantage to have the taste for bitter compounds or it's a disadvantage. And I was hoping that I would give you a perfect answer, but really the answer is not perfect, because this is a study from the Kangra Valley in India, Himachal Pradesh, India. And this is in adolescent girls. And what they found is that the tasters, which can perceive bitterness, had significantly lower body weight and fat-free mass. So, in other words, this is a little paradoxical. So here are the tasters. The tasters are lower in height, and they are higher in fat-free mass. Sorry, they're higher in body fat mass. In other words, they have more fat and they are lower in height. Something that is not desirable, I would assume. And this study, people said that the reason for this is that if you were a non-taster, you ate more and you ate better. On the other hand, if you were a taster, you went more for sugary food and not bitter food, and hence you had more fat mass and you were lower in height. So at least that was their conclusion. On the other hand, this study comes with a diametrically opposite opinion. This is a study of PTC tasters among obese individuals. And in this, they divided people into normal weight group. There was an underweight group that I did not show, an overweight group, and an obese group. So you would expect, if it was following the same pattern as the study from India, that these results would not be there. But in here, what was found is that having the taste for bitter compounds was good because you are more likely to be normal weight. And if you were a non-taster, the signaling from the bitter compounds that would reduce your food intake would not be there, and you're more likely to be obese because non-tasters had a higher incidence. In other words, amongst obese people, the number of non-tasters was much higher compared to the tasters, compared to the normal weight group. So in other words, lacking the sensation of bitter taste through taste receptor 38, there was a higher association of that in the obese individuals. Now, I'm going to touch on a subject that many people have asked me about, and I am by no means an expert. And almost all of this presentation is hypothesis generating, thought provoking. There are no definites that I'm giving you. But what I wanted to explore was low calorie sweeteners. Are they good, bad, or neutral to our health? So this is a study of an artificial sweetener, sucralose, Splenda, on gastric emptying, and incretin hormones are actually GLP-1 and GIP. So on incretin hormones and insulin. So what happened is that in this study, they gave 50 grams of sucrose or an equivalent sweet sucralose, which was 0.4 millimoles, or a 10-fold higher concentration of sucralose to see whether blood glucose goes up, whether insulin goes up, whether the satiety hormone GLP goes up, and whether the insulin releasing hormone GIP goes up. What was found is that when you gave sucrose, sugar went up, insulin went up, GLP-1, the satiety hormone went up, and GIP went up. But when you gave sucralose or saline, none of these things happened. So in other words, this study is arguing that sweeteners don't increase sugar, don't increase insulin levels, don't increase the level of satiety hormones, and hence they are good. This is another study by John Peters, and it says low calorie sweetener use and energy balance. And John Peters is arguing, based on previous studies, that the association between low calorie sweeteners and obesity, which is BMI increase in observational studies, is actually due to reverse causality and residual confounding. So he says, all the studies that are telling you that taking low calorie sweeteners is bad for you is wrong. And he's talking about randomized controlled trials that consistently show a benefit of low calorie sweeteners. And consequently, this is simple behavioral change for people to control their weight. So this is quite endorsing the use of low calorie sweeteners. And I think I was born a skeptic. I mean, I really don't trust a lot of studies that are put in front of me and I explored. So I decided to explore this a little bit. And the University of California, San Francisco has a lot of email and other documents between the university and the food industry. And this group looked at potential confounding factors or front groups that put out these articles to make us think that low calorie sweeteners are beneficial. So at least there is a bias. And when you looked at these documents, I found that Dr. Peters was in close contact with the industry people, which is the IFIC, International Food Information Council, which is an industry group. And consequently, I am concerned about the lack of neutrality and potential bias in what John Peters, who put out this study, is showing, because he's not only mentioned in one area as somebody that IFIC should rely on, but also mentioned in a few different areas. In addition, in one of the documents, it's saying that this person was quite saddened, sickened by some top experts in the field, because an Israeli study that was questioning the use of low-calorie sweeteners, and it's trying to say it's full of holes, is not reproducible. Why did this Israeli study got so much publicity is an indication of extensive animosity that exists against the food industry. So in other words, it's putting down the study. So this was the study that is measuring artificial sweetener toxicity using a bioluminescent assay. So you use bacteria that light up if there is toxicity from sweeteners. And this study was actually questioning that artificial sweeteners are linked to cancer, weight gain, metabolic disorders, type 2 diabetes, and alteration of our gut microbiome. And moreover, these artificial sweeteners are ending up in our environment, in our surface water, in our groundwater and drinking water. So this is not just of concern to people consuming it, but concern to the overall population as a whole. But we do know at least that this is a fact, that artificial sweeteners do interfere with the hedonic center, the pleasure-seeking centers, and promote food intake and promote hyperphagic behavior. And that withdrawal of artificial sweeteners creates craving. So there is well-documented literature that these things happen. Whether they are linked to cancer, weight gain, metabolic disorders, type 2 diabetes, at this stage is unknown because of the lack of proper studies and a lot of bias and propaganda from the food industry. So these are the last two slides in which I'm going to summarize what I have discussed with you. I think that most humans will require interventions to reduce hunger and improve satiety. Without that, at least in my clinical experience, and I've been doing this for, in other words, I've been talking to people about nutrition for almost a decade, I feel that most of us are designed to overeat. Sugar and refined carbohydrates activate both the chlorogenic, in other words, the homeostatic, energy-driven centers, as well as the hedonic centers in our brain, and make us overeat. I think that understanding hunger and satiety hormones, in other words, understanding basic biology of how hunger works, is important for our health, because then we can modulate our behavior. One of the primary reasons I put this out is to put out, and I want to get this question asked at the end, and we'll have some time, that bitter tasters are perhaps useful in improving satiety. Now, low-calorie sweeteners certainly activate hedonic centers and perhaps increase hyperphagia and obesity, cancers, diabetes, and alter our gut microbiome adversely. And my strategies to promote satiety to you is the following. Number one, fasting reduces the size of the stomach, and it promotes early satiety because the distension of the stomach leads to fullness and you stop eating. Perhaps something that I didn't get into is that animal protein perhaps improves satiety and satiation better than fat and carbs. Carbs definitely increase hunger and reduce satiety, but I think an animal protein meal is perhaps the best to improve satiety. I think we all should consider avoiding sugar and refined carbs, which is basically the message that most of our community members give. And if you looked at my previous presentation on calorie restriction, I think that we need to design a strategy in which we stop eating when we are 70% full, because it takes about an hour and a half for ghrelin to come down. And for the satiety hormones, which is GLP-1, CCK, and peptide YY to kick in and tell the brain that you have eaten enough. I want you to consider bitter tastants in order to improve your health and improve satiety. I think bitter tastants should be consumed from the beginning of the meal so that they have time to act rather than at the end of the meal. But I would like to get, if anybody has personal experiences, and maybe even put out a poll. I think that low-calorie sweeteners are bad news, but I don't have literature and enough information to back it up conclusively. Bariatric surgery does some of the same things to reduce hunger and improve satiety, because bariatric surgery gets rid of ghrelin. Bariatric surgery is designed, especially with gastric bypass, to bypass the duodenum, reduce insulin secretion, increase the secretion of GLP-1 and peptide YY, which produces satiety. But I'm not endorsing bariatric surgery by any means, because it comes at a high cost of nutrient malabsorption, esophageal dilatation, perhaps lung infections, because you have a lot of reflux that will reflux stomach contents into your lungs, and also dumping syndrome, among many other things. So I'd like to thank you, and I hope to see you next month. And so long and goodbye for now.